Hey guys, my name is Alexis, and today we're going to be doing a genetic profiling lab. The purpose of this lab is to isolate DNA from buccal cells, or cheek cells, by using items that you could find around your house. You will be able to show that by using your DNA and biotechnology, you can develop diagnostic tests for diseases and determine which medications to use. You will also be able to identify and explain the purpose of each step in the procedure as well as the possible results. Before we begin this lab, be sure to familiarize yourself with the following key terms in order to understand and be involved in the lab experience. If you're not familiar with these terms, please pause the video now and research them. The underlying principle of this lab is human diversity. Our genomes are made up of 3 billion base pairs, 99.9% of which are the same in all individuals. It's the other 0.1% that make us different and make it possible for the practices such as forensics to identify individuals simply by their DNA. Today, you'll be taking a sample of your own DNA, isolating it and replicating it. You will then run a gel electrophoresis of yours and a classmate's DNA to observe the differences. To carry out this procedure, prepare or collect the following materials: 1 50 milliliter tube containing 2 milliliters of water, 1 50 milliliter tube containing detergent, 1 50 milliliter tube containing 95% cold alcohol, 1 sterile 1.5 milliliter microcentrifuge tube, 1 sterile 0.5 milliliter microcentrifuge tube containing 25 microliters of 2x PCR mix, and 1 P1000 pipette and blue tips.
In order to obtain the buccal cells, drink water and swish it around. Swishing the water around helps dislodge the buccal cells from your cheek. After swishing the water around, spit the water into a tube that has detergent. What is the purpose of using the detergent? Pause the video to answer the question. Cap the tube and invert the tube several times. Make sure the cap is on tight. Open the tube and tilt the tube 45 degrees. Using a P1000 micropipette, drip the cold alcohol down the side of the tube. Make sure the tube is tilted during this process. A cloudy area should appear where the two solutions meet. This cloudy area represents the DNA. Using a micropipette, transfer the DNA to a tube provided by the instructor. Using a microcentrifuge, spin the DNA at a top speed. A pellet DNA should form. This pellet may be hard to see. Place the tube up to the light in order to observe the pellet if needed. Remove the supernatant and allow the pellet to air dry. Using 250 microliters of sterile deionized water, resuspend the pellet. Mix. Set up a PCR in a 500 microliter tube using 25 microliters of 2x PCR mix, sterile deionized water. DNA and 2 microliters of VNTR, which is variable number of tandem repeats. Using the thermocycler, run the PCR. Finally, prepare and run gel electrophoresis.
Your results should look similar to the picture on the screen. The lane on the right is the ladder. The ladder is used as a reference for the size and quantity of DNA fragments. The first 3 lanes to the left belong to student A, and the lane in the middle belongs to student B. You will notice that the 2 students have mini bands that were almost identical. However, student A also has bands that are not present in student B's lane. This is a good representation of the variations in DNA between 2 individuals.